The overall goal of this imaging and analysis method is to generate an easy-to-set-up workflow for the automatic segmentation of any fluorescently labeled slide-mounted tissue sample.
This method can help answer key questions in the biological sciences field about how much of a given protein is found within a specific area of a tissue sample of interest. The main advantages of this technique are that the time-optimizing protocols for automated segmentation can be saved, allowing this method to be applied to subsequent fluorescently labeled slide-mounted tissue samples. We first had the idea for this method when we needed to quantify immune cells in brain tissue at a high magnification.
After importing the journals, open the Plate Acquisition Setup dialog box and open the Objective and Camera tab. Set the magnification to 4 times, the camera binning to 1, and the gain to 2. Open the Plate Slide Scanning tab and input the appropriate experimental settings. Under the Sites to Visit tab, fill the wells uniformly with the sites and open the Edit Plate Bottom Settings tab to enter the appropriate experimental values. After saving these settings as setting A, open Objective and Camera tab and set the magnification to 40 times, the camera binning to 1, and the gain to 2. Confirm that all of the settings under the Plate Slide Scanning and Edit Plate Bottom Settings tabs are the same as entered for setting A and open the